Welcome everybody to today's video. In today's video, I'm gonna be explaining the process of translation. This is the part 3 of the series of protein synthesis. So part 1 was about the main ideas you need to know in order to go deep into the details of this interesting process. And in part 1, I discussed the transcription, which was the first process, and then I'll discuss now the translation, which is the second process, and part 3 will be the last part in the protein synthesis. Are you ready? Let's go. So, what is translation? Translation is the process in which genetic information carried by an mRNA, the messenger RNA molecule, is decoded in the ribosome to form particular polypeptides. So, we need to understand that translation is simply uh, translating the information that the messenger RNA had sent it to the cytoplasm, to the cytosol, in order for the polypeptide to be formed. So we talked about the transcription in the last video, and we said that in the DNA we have information. This information, okay, will lead to the formation of a polypeptide. Each gene or each part of the gene codes for a certain polypeptide. How? Because each three nucleotides will code at the end to a certain amino acid, and I will show you how. So this mRNA, the messenger RNA, what it did is that it only got the information from here and sent it and went to the cytoplasm, cytosol, to the ribosomes in order to translate these information into an action, into amino acids that in turn will form the polypeptide chain which we want at the first place. So a simple recap is that we have the DNA with three. I only gonna do the example on three nucleotides, but it's of course more than three. Let's say that this one is the coding strand. If you don't know what is the coding and the template strand, go and watch the last video. It will help you a lot. It's about the transcription. And please don't watch only the translation video because it's a series. It has to be watched like all the parts for the ideas to be interconnected. Okay, so watch 3 videos by order and you will understand the whole process of protein synthesis. Okay, so let's now continue with translation. We said that this is the three nucleotides in a coding DNA strand, and these are three nucleotides on the opposite strand, which is the template strand. The mRNA will be formed here, okay? And it will have also three nucleotides complementary to these, okay? And we call these three nucleotides a codon, okay? Now, the mRNA will get these three nucleotides or the codon, which is the information, which is basically the information but in another language. It will go out of the nucleus and to the ribosome here. See, these are three nucleotides of the mRNA, messenger RNA. These are the codon. And we will see the tRNA, the transfer RNA, with also three complementary nucleotides. We call them anticodon. It will be attached to the anticodon from one side and from the other side to the amino acids that is complementary to the anticodon, which is complementary to the codon, which is complementary to the here to the template DNA strand or template DNA codon, which is complementary to the coding one. So initially, the information started in the DNA, then sent to the cytoplasm. I'll use another color. Sent to the cytoplasm, and then from the cytoplasm and the ribosomes to the tRNA, which attaches with the amino acid that is appropriate to the information we inserted in these ribosomes. After this explanation, I just want to make sure that I didn't miss anything. This is the nucleus. Here is the transcription, and here is the translation. The messenger RNA will go here to the small ribosomal subunit and the large ribosomal subunit. After these two subunits attaches with the mRNA, the process will begin, but not until the tRNA will arrive with its anticodon and its amino acid, which is always the first amino acid is methionine. Okay, methionine. We may call this tRNA an initiator tRNA. Is the tRNA the transfer RNA that has the methionine and the first anticodon, which is complementary to the first starting codon. Here, what you need to know is that the starting codon, the AUG codon, is always the starting codon. So when you hear the starting codon, the first thing that should pop in your head is the AUG. AUG. So what is the anticodon? It will always be UAC. Okay, complementary to the AUG. That's why methionine will be always the first amino acid in the polypeptide chain. And by the way, it will detach at the end. After the polypeptide will be released, the methionine will not complete the journey with this polypeptide wherever it will go. It will detach from it, okay? And of course, here I'm not focusing on how A here and then we have the U complementary to the adenine, we have the uracil. I'm not focusing on this stuff. I'm just trying to convey the role of these nucleotides in the movement of the process. Okay? Because here the starting codon is the codon that starts the process, and here the anticodon is the codon is the nucleotide sequence that will form the methionine, right? And then everything will move according to these letters. I'm not gonna talk about how AUG, guanine, cytosine. I'll not talk about that. I'm just focusing here about translation in this video. Okay? And as I said, this is for the last time, this is the transfer RNA. Here we have the anticodon. Here on this side we have the amino acid that attaches. Not only the initiator tRNA has this structure, every tRNA has this structure, but the difference is that not the methionine, the amino acid that will attach in this side will be another amino acid. There are other amino acids that can be present. Okay? And in fact, how does the correct amino acid attaches to each species of tRNA? Okay, how does methionine find this site? How does this methionine go to this site and bind to it? By these synthetase enzyme. Okay, what is it called? Amino acid tRNA synthetase. Okay, this enzyme is the enzyme that is involved in the process of this amino acid attaching to the transfer RNA. And after I gave you everything you need to go into this topic confidently, I will start talking about the steps. We have initiation. This is the first step. Elongation, the second one. And termination, which is the third one and the last one, by the way. Okay? What happens in initiation? Initiation is like the beginning. How does it begin? Okay? What is the mark that begins the process? Okay, when we have small ribosomal subunit, large ribosomal subunit, check, check. The mRNA is here, check. An initiator transfer RNA carrying the amino acid methionine, here it is, so check. So these four things have to be present in order to move to the next level, the elongation. Now, the elongation begins. In elongation, amino acids are added to each other and bonded by peptide bonds to form the polypeptide at the end. All right? This is the elongation. And it also has three steps. We have codon recognition, peptide bond formation, and translocation. Let's begin with the codon recognition. The anticodon of an incoming tRNA, so let's say this is the anticodon, binds with the complementary mRNA codon. So binds with this one. And what do we mean by complementary? We mean the letters here complement these. Okay? So we can't, so if we have this tRNA, for example, A and then G, C. Is this anticodon complementary to the CG? No. And why? I'm not gonna explain why here. It's a letter thing, okay? But of course, it's chemistry, okay? It's chemistry. But here, I'm not explaining that. I'm not explaining the letter thing. I'm just gonna say that the GGC is the complementary letters of the CCG. Okay, so C should have G in front, C should have G in front, and G should have C in front. Okay, but AGC is not complementary, so it will not go here. That's why we said that the anticodon binds with the complementary mRNA codon, okay, in the A site of the ribosome. An important thing to know that initiation starts here. Here in this site, P site, not the A site, the P site is the site where these four things that should be present will be present, okay? So in the P site, but then the elongation, so the second, third, fourth transfer RNAs, not the initiator of transfer RNA, will first go to the A site and then we'll move to the P site in the translocation, which I'm going to talk about, okay? So here, okay, look, we have already here a polypeptide chain. So this is a little bit misleading. I'm just gonna draw here simply. This is the large unit, this small unit. And I'll draw this is the tRNA. I know it's not like that, but I'll draw it like that. And this is methionine, okay? This is the tRNA. And this is methionine. It's just a simple drawing. Here we have the P site, here the A site, and here the E site. Okay, so first of all, we have these four things, and then the initiation is over, then we have the elongation. In the elongation, another tRNA, it's not the initial one anymore. It has the anticodon. I will go with red this time. The anticodon complementary, of course, you know this is the mRNA, complementary to the codon of this. And the anticodon here, wherever it was, maybe ACC, will code for a certain amino acid here. So I'll just put a random number S, okay? It will go here in the A side, okay? But here they are like drawing a picture of a advanced stage of the process of elongation. But just understand that this polypeptide has formed because of the repetition of this process I just explained, okay? So this is the anticodon. It will go to the A side, complementary to the codon, and we'll have this amino acid, the corresponding to the anticodon. By what enzyme? We said this one, look, this one. Okay, the synthetase enzyme. This is how you write the name. Aminoacyl tRNA synthetase. Okay. Now, how this amino acid, I will call it L, okay, will bind to this polypeptide chain. Is it from this side or from this side? Look, what I need you to imagine. I need you to imagine that. I'll draw one here. And by the way, this is the peptide bond formation. I'm going to explain now. The growing polypeptide bound to the tRNA at the P site. So if you gonna go back to the first picture, you're gonna notice that it is in the P site, right? Is transferred to the amino acid carried by the tRNA in the A site. So it will be transferred to the L amino acid here. I will show you how and a simple way of remembering it, okay? So a new polypeptide bond is formed. Let's say this is the large subunit and this is the small one, all right? Here we have the tRNA in what? In the P site. And here we have the tRNA in the A site. This one is the new tRNA, new, okay? And this one is the one that came before. And by the way, if it was more than methionine, like MEC. So certainly we have a tRNA here that had left already in the E. And I'll talk about this leaving thing later, okay? So this new tRNA will have also an amino acid. I will go for orange here because here I'm focusing on the point of the amino acid, the new amino acid attaching to the polypeptide chain. How does this happen? I'm gonna tell you. Okay, see, look. Here, this amino acid or this polypeptide chain will detach. Let's say that this is a girl and this is her father or mother. So let's say it's her father. So it has a balloon. It grabs the balloon, but suddenly it lost it, and the balloon is released far. So it lost it, but here the father was able to grab it. See, look. See here in this picture. See how the balloon was lost? Originally, this was the balloon. Look in this picture. PRO was the first one here, but it lost it, and then this one managed to catch it. And the hand of the mother or father, is the new peptide bond. Okay, so that is how the polypeptide is formed by the repetition of this process. So not the new amino acid is the one to move or to detach from its tRNA. The whole polypeptide chain that was before in the P is the one that to detach, and then it will attach to this new amino acid. That's why the methionine is at the end, because first of all, you can imagine that it was here, right? And then we had, for example, E. So then it detaches and will form there. Okay, so and then we have S. So this will detach and will form attach here. I'll do it another time, the last time. Here, let's say we have N. This is a new amino acid. Now this, let's go for purple. This will detach and attach here. That's why methionine, the letter M, methionine, MET, will be always the last one. Okay, and the new ones will be attached from this side. All right. And don't worry, just at the end, I'll do like 1 minute revision of what we said in order for you to understand in a simple way and a complicated way, and then you can be able to visualize what is happening in your mind. And I'm telling you, Visualizing it, visualizing the process on your own, in your mind, without watching the video ready for you, is really better than watching it, like only watching it and then you think that you understand the process. Your brain has to work. Your brain has to ask questions and to find answers in order to find the picture of the story. You need to visualize it on your own, depending on the information that you will get in this video. And after you visual, I'm not saying don't watch the video. The video will be really, really helpful. But when you visualize it first and then go watch the video, visualize it by yourself in your own way. Okay. And then when you watch the video, you can edit your visualizing. Okay. But don't get the visualizing from another's video directly. Okay. That was an advice. Let's move to the translocation. Okay. And many people think that here, when the tRNA in the P site becomes in the E site and the A1 becomes in the P, many people think that the mRNA is the one sliding or the tRNAs are the one jumping, but it's not the case. The large unit subunit and the small subunit are the ones that are moving. So these, the ribosome is moving. The mRNA is it's in its place. It's not moving. So when these are moving, just now you need to visualize it. See, look, now this is in P. This is in P. And this is an A site, okay? When this will move here, I will draw the new one. It will become like this, right? It moves one codon at a time, okay? So now this becomes the A site, this is one the B, and this one the E. So here, this tRNA I'm lining now will become in the E, whereas it was initially in the P. And the one that wasn't in the A site will become in the P site. And then here the A site will be empty for another tRNA to join the party. Okay? That's how translocation happens. An important thing, as illustrated here, when the tRNA in the P site that was in the P site becomes in the E site, we said that this P, before the ribosomes move, it will lose the polypeptide chain. So now it doesn't have an amino acid. So we are okay with it going. Okay. So after this tRNA gave its polypeptide chain in order to continue building up the polypeptide chain and for it to become longer, after that happens, this is not beneficial anymore. So when the ribosomes moves like this, it's just saying like, hey, tRNA, you can move to the E, to the exit where you can go. And I need to make a place, the A1 here, for a new tRNA in order to continue the process. See, look in this picture. Here, I'll use blue, for example. Here, the A site is empty now. And this one that was in the P site actually became in the E and was released. Okay. This is the tRNA that was released. And then this tRNA in the P site will lose this polypeptide and another tRNA, whatever, with a new, let's say N, new amino acid will go to the A site, and then after this polypeptide deattaches, it will attach to this amino acid, and the polypeptide will be longer a amino acid from what it was before this amino acid joined. Okay? Don't worry about the process. I said I have a 1 minute that will discuss everything we talked about, but in a simpler way. It is important to see it in the complicated and in simple way in order to understand it. Okay? So now the last part and the last step in this process in translation is the termination. I said that the nucleotide sequence, these letters, it's true that they are really tiny, but they have a huge effect in all processes that involves genes and doesn't involve gene because gene is involved in everything. It decides everything. And what are these genes? They carry information in the form of these letters, tiny letters, which are conveying messages, uh, doing some roles. They can do anything. They are like when you are studying biology, for example, you have like the book that where you hesitate if something was true or wrong, you go to it. It has the rules. Okay, so these are the rules that everything in our body moves according to. Okay, so here now, when we have the stop codon, when we have UGA, or it could be UAA or UAG, either three of any one of these three sequences, or let's say codons, are capable of stopping the process because there's no amino acids. Because when we are gonna get the anticodon for each of these codons, okay, I will do it like quickly. It will be here U, so it's A, C, U. So these are the complementary anticodons. The tRNA should come here and have one of these three. Okay, in this case, UGA, this one, it should have ACU, but there's no amino acid that the ACU, we said that the anticodon is the one that codes the amino acid. There's no amino acid that the ACU codes. That's why there's no binding here. So now the release factor bind in the A site. Okay, so I will just go back here and then there. Now we have APE. In the E side, it will exit, all right? Let's say, for example, we have another tRNA that came here and with N, and it got this, and this came here, and then this came here. But how? These are not moving. The tRNAs are not moving. The one that is moving is the ribosome. Okay? So the sites are the moving things. So when the sites move, of course, the tRNA will be obligated to change their site. They don't move, they stay in their places, but the sites are the one that move. Okay? So now after this movement, this one will be without amino acid. In the exit, it will go out. And this one will be here. And if now we reach the either UAG or UAA or UGA, this release factor will bind in the A place, and this will signal the end of the termination, the end of this process. Okay, and what will happen then? The release factors triggers the ribosome subunit to separate. So now the subunits separates. There's no attachment with the mRNA anymore, and the mRNA will be released. The messenger RNA will be released, and the new polypeptide will be released. And now we will benefit from this polypeptide that the DNA informed the RNAs to do. So let's say that the DNA is the one with the information, is the one that gives the ingredients, and the RNAs are the chefs, are the one that cook the ingredients. Imagine it as you like. The DNA has the information. RNA just like use these information to form the result that we want from this entire process, which is polypeptide. Why? Because polypeptides in the first part, in the part one, that is why part one is important. Don't skip it. Part one, it is important. I told you how the polypeptide is the units of the protein. How this is how we synthesize proteins, okay? And now I'll share with you a quick summary, and it is really important since it will consolidate your understanding. Believe me. Now, nothing happened. We have a nucleus, we have DNA. Now, the mRNA went out of the nucleus, or let's use the present here, goes out of the nucleus with the information that it transcribed it already. Okay, so here we have the cytosol. went out through the nuclear pores. Okay, here it goes like this, the mRNA with the nucleotides, and it will attach with the large subunit and the small subunit. All right, you need to know that we have a P site, an A site, and an E site. Okay, and these sites are from the ribosome. So when the ribosome will move, the sites will move. This is important. Okay, I'll just do like this to separate the sites. You can say that P is the site where the initial tRNA, transfer RNA, will get in, okay? And then all the other RNAs, tRNAs, will go to the A, okay? Now, we have an initial tRNA, and everything is here. We have the codon, which is the starting codon. Every time it's AUG, with the anticodon, and every time it's UAC. We have these. We have the large subunit, small subunit. Then this will go to the P, and since it is UAC here, the anticodon is UAC here, will be M. Okay, so I'll draw it inside. Okay, and now the initiation is over. We have the elongation. How does the elongation happen? We have another one, tRNA. Let's say, for example, here we have CCC for simplicity. Okay, here we have CCC, and this is the codon. Now, the tRNA with the anticodon GGG will come here. And since it is GGG, here we have a certain amino acid, wherever it was. Let's say you are not required to memorize these, of course. But there's like a drawing that will be provided in order to figure it out in any exam, okay? But it's good to always ask your doctor about whether it is required or not, okay? So let's say here we have N, for example, the amino acid. It will go to the A side, and this is, by the way, the elongation. It will go to the A side, okay? I will just draw it in the A side. And with the anticodon. Okay, after that, what will happen? We have the translocation, as we said, right? These will move like this, the ribosome. So now we don't have these now here. And since the ribosomes move, right, it's one ribosome. The ribosome move, the sites will move, right? So the ribosome will become like this. It moves one codon at a time. This is the large subunit, and this is the small one. And since it's moved like this will become the E site, and this one in the P site, and this one in the A site. What we have in the A site? Nothing. Do we have anything? Can you see anything? No, because it's an empty space to another tRNA to arrive. And of course, we have a codon. It moved, so I will erase the arrows. The codon, let's say it's AAA, for example, just for simplicity, okay? Adenine, adenine, adenine. So now, what will happen? Okay, I cleared the picture in order to draw a better drawing. In the A site, we had the new one that arrived. We said it was N, I think. We don't care now. We are just saying a certain amino acid according to the anticodon here, which is according to, it should be complementary to the codon here. So we said CCC, this should be GGG, okay? Now we need to form the polypeptide, all right? How will this happen without a peptide bond? Okay, so here a peptide bond will be formed. How will this happen? I said the balloon will be released like this, and this one will catch the balloon. So when it was released, this one coated. So this one will be not here anymore. It will be released. It will become here, bonded to the amino acid. Okay, so now the ribosome realized that this tRNA, what is it doing here anymore? It doesn't have an amino acid. It will not contribute to the process. So we have to empty a space, and no tRNA will go from here, will come from here, because the process will be like this, okay? So we have to empty the space in order for a new tRNA to arrive, in order for the polypeptide to elongate. Okay. So what does the ribosomes do? Right, it's one ribosome, but actually they may be more than one ribosome sliding on the same mRNA. But we are not into this now. We are just describing or explaining how it works in translation for one ribosome. So when it will, the ribosome will slide like this, the small and the large subunit, the sites will also slide because they are basically in the ribosomes, you know. So when it will slide, the sites will slide as well. Okay, so since it slides, I will erase this one. Okay, and the sites, I'll erase the sites because the sites will change. And the ribosome slide one codon at a time. So one site at a time. So it was here, it will slide one, two, three, so slide one codon till here, and it will form this. Then, like the ribosome, you can say it can contain three codons, okay? And this is the small one. Now what happened? The sites had changed. I erased the mRNA. This is the mRNA, okay? Now this is the E, this is the P, and this is the A. Take it as a rule. When the tRNA arrives at P, it will leave. So it's over. This one, this tRNA, have left. Okay? But of course, the codon will not leave because it's a part of the mRNA, the messenger one, which is still here at its place. Okay? But now what will happen? Here, the A is the site that receives the new tRNAs. Okay? So now, let's say a new tRNA will arrive. According to what? According to the codon here. So let's say it was A. So the tRNA with the codon U, anticodon. This one, the anticodon, and this one is the codon, will arrive at the site A. And the U anticodon will face the AAA codon, okay? And he will attach a amino acid according to the UUU. So maybe the amino acid that is appropriate to the UUU, let's say it's C, okay? Of course, there are names, okay? But you are not required another time to memorize them. Now, let's say it arrived. I will erase this. It arrived at this level, at the site C. Okay, so as you can see, it's at the A site, in the A site. Now what will happen? It will happen again. This balloon will be lost like this, and this will catch it. Okay, so now we can assume that we can visualize now that this balloon will be gone. Okay, I'll draw it again. Do you know why? I believe it was N, whatever. But let's say the orange one and the yellow one. This is N. I will draw it just to show you how it is released like this. Like this. So I'll draw it here before the bond happens. It will be like this, right? Same. See, the same. But here the different thing is that this C, the amino acid in the A site will catch it. So here we have now two peptide bonds, and the amino acids, the three ones, are bonded now together and formed a longer polypeptide. So that now we have a 3 amino acid polypeptide. Of course, this is gone without the arrows, without anything. And now this is the shape. Another time, I will repeat. Look, here, the ribosomes, or the one ribosome, realize that this is an empty one. Okay, let's empty a space in the A site and push this to the E site because we need it to go. That is why the ribosome, one more time, not one more time, of course, it will continue to move until the process will finish, will stop after termination, as we said, because of the stopping. Everything, stopping, initiating, everything is because of these letters, okay? So because of the stopping codon, all right? So now let's draw it. Here it moves like this. So the sites will change. Everything is at its place. Only the ribosome will slide. And by the way, this will require GTP, but let's not talk about this. This is the big, large one. And it was like this. Now it moves one codon. So it skips this and it is like this now. I'll just fix this to be more real. Okay. But now when we skip this one, we have another one that will appear here. Of course, we have this small subunit. Okay. Let's say it was CGG. Okay. So now we have a new, I'll draw it here, for example, a new tRNA, which has the anticodon complementary to this codon, which is GCC, will go to the A site. Okay. And for another time, we have here, it will attach to the amino acid that is appropriate to this anticodon, which is complementary to this codon. So it's all like mixing together codons, and the amino acid will be appropriate to the first codon in the sequence, which is the codon in the DNA. Since the DNA is the first thing, then the mRNA, which is the codon, then the tRNA, which is the anticodon, and then we have the amino acids. So the whole story begins from the DNA, and this is, I think, the 15th time I'm mentioning it because it's really important to understand the role of DNA in protein synthesis, because without DNA, there's no protein. There's no protein at all. Like if we have only RNA, from where they will get their information in order to work and build the polypeptide, you know? That's why. Okay, so now this tRNA, let's say the GCC will result in the binding or attaching of the R amino acid, for example. Now this will move here. I'll draw it. This is the R, and this is the anticodon. And for the last time, I will tell you this story. The balloon will go. I wish I had some space here. You know what? I really care. I really want my picture to be clear for you all guys. So I will redraw it. Now we have some space here. I was going to talk about the balloon story, but before, I'm going to ask you a question. Try to answer it, okay? And this will reflect the understanding in the video. What did we miss? What is the step that should happen before the balloon story? Okay, guys, you can pause the video. If you didn't know it, it's okay. It's okay not to know something, but what is not okay is not to find the answer for the thing that you don't know, okay? So, when the ribosome slides, we said the sites will change place, okay? So here E, P, and the A. EPA will move. Therefore, the tRNAs will not still be in their places, in their sites. And I told you, when the tRNA arrives to the E site, it will immediately go away. You are not beneficial anymore. You don't carry any amino acid that will contribute in the polypeptide. I need to empty a space in order for another tRNA that has an amino acid to arrive and to contribute in the process, as I will illustrate now and explain to you in the balloon story. Now, this balloon will be released like this. I will draw it here. Okay, so now since it is released, so see it look the same. It is released now. This is gone. This is the same as this one, but the RNA can catch it before it go away. So this is a new peptide bond. And now, by this process, and the repetition of this process again and again, the polypeptide will lengthen, right? What will happen next? Think about this. Okay. It's okay if you don't know again. After this, we said that we have this one. It doesn't have an amino acid. So the story of emptying a space and letting some unuseful let's say, are useless, tRNA to go away for now. So this one, the ribosome will slide now. This will become here in the E site. It will go out. This will become here, and a new tRNA will join here according to the codon here. And now the balloon story, and now the polypeptide has 5 amino acids, and this will continue until we reach the stop codon. So now we said that after the balloon story, after this catches the balloon, this will slide. So the tRNA with the polypeptide will go to the P side, right? And here the one without the amino acid will go to the E side, right? Okay, let's draw that. This is in the E side, and the one with the polypeptide chain is in the P side, and the A site is empty for another one to arrive, right? As we said, this is the, okay, wherever names of these. But the important thing is, and I'll draw it in the different color, that the last one is methionine, as I said, okay? Here, the process will repeat itself until this ribosome will move one codon to the right until it reaches the empty site A with one of the stopping codon. What are the stopping codon? UAG, UGA, or UAA. Is that clear? So any of these three codons can stop the process, can stop the elongation, and then we have the termination. So when this ribosome arrive at the point where the empty space A has one of these, what will happen? We have the release factor, it will bind here. Of course, this will exit. The first thing, any tRNA that will arrive here, it will exit. Now the release factor will bind here, and then the subunit will detach, and the mRNA and the polypeptide will be released. Good to see you all at the end of this relatively long video, but you can do it, guys. You're amazing because you are still watching till now. Don't forget to watch the whole series. It is important to watch the whole series, 3 parts, part 1, part 2, part 3, in order to have a complete understanding of the process of protein synthesis, okay? And if you want me to do a video regarding the letters, these bases that I illustrated in this image here in front of you, G, C, adenine, cytosine, and these stuff, please write this in the comments so I know that people really want that video. And thank you for watching this video. Don't forget to hit the like button and subscribe for more. See you in another video.